Hi, I'm Dr. Jeff Habel. Today I'm going to talk to you about a poster that we're presenting concerning an automation of a monoclonal antibody workflow. And so we're going to be doing this on an NGC system. And what makes this unique is the fact that we're going to combine both a tandem purification with a multidimensional purification. And here on the poster, we see the fluidic scheme of the system that we're going to use. And again, we're combining tandem, so two columns in line together, with a multi-D solution. So when the protein, the antibody of interest is eluted, it's going to come back and be put into our loop for storage, and then loaded onto the next tandem for continuous purification. And so traditionally, what a scientist would do is they would do each one of these steps individually. So they might do a protein A column, and then because of the way that the protein's eluted off of protein A at low pH, they would either neutralize it or dilute it instantly, then load it onto an anion exchange column in flow-through mode, and that's to bind out the nucleic acid and host cell proteins. And then bind in elute mode for cation exchange is very common. And then in this case, we're going to do some analysis with some analytical size exclusion chromatography. So what we've managed to do in this application is take this entire workflow and then automate the entire process. So we still do the protein A. Desalting takes the place of our neutralization, so it's instantly neutralized. Then we're going to store that inside to a loop, and then that loop is going to go through an anion exchange column, again in flow-through mode, and then directly onto the cation exchange column and bind in elute. When we elute off the cation exchange, again, we go into a second loop for storage, and then a small portion of that volume is sent through analytical SEC for analysis, and the rest from the loop is just sent to the fraction collector. And so the interesting part about this whole process is to generate this single automated method, you're actually using all the information that you gain from the optimization of your traditional purification. So it's really no additional work for the scientist involved. So for example, here we have, we see the ion exchange tandem optimization. And so what we do is we do a simple pH screen to try to figure out which is the best pH for us to use for loading the sample through the anion exchange column. Because again, our protein of interest isn't going to bind. And so what we want to do is maximize the percent recovery, and you can see as we increase in pH, as you would expect, more and more of our target protein binds, which is exactly what we don't want to have happen. So that's why we select a low pH in this case. And then for the cation exchange column, where we actually want to bind, we bind, and then here we're doing a percent B scout, so we're trying to find the best elution condition. And again, you can see as expected, we get tighter and tighter peaks. You can still see this little bit of a shoulder, which we'll then analyze. And so taking this information from the optimization, you can then combine that into the automated method. And so in the end, we do size exclusion analysis, whether that's individually or through the automated portion. And we can see in the blow up that we're actually separating out some high molecular weight aggregates from our monomer peak. And finally, we put all of this together and we demonstrate that with a single button push, you can do this entire protein purification workflow through the protein A and desalting. Then it's saved into a loop. We go through anion through flow through, cation is bind in elute. We save this peak into a second loop, put a small portion in through the analysis, and save the rest. And typically, scientists in process will analyze for host cell protein contamination. And we just have a simple table showing that whether you do this sequentially or in the automated tandem multi-D combination, you basically have the same answer at the very end. So whether a scientist does this by hand or in an automated fashion, you get the same result. And the best part about doing the automated portion is every time you press the button, you're going to get the same result in the end. So it becomes very reproducible and minimizes human variability. For more information about this process, please go to Bio-Rad.com.